There are a variety of factors that cause mutations to happen in a cell. We have already talked about DNA replication errors and how DNA replication process itself has some proofreading built into it. Our polymerases can do some proofreading. So that's one way that we can develop a mutation. There are a variety of other factors by which a cell can acquire mutations as well. So we may acquire an insertion or a deletion, and this can happen by a process called strand slippage. So in this mechanism, basically what happens is that we get an incorrect base pairing happening, and it leads to a bulge or a nucleotide sort of popping out. And depending on which strand this happens on, we can end up with an insertion or a deletion. So this often happens when you have a set of nucleotide repeats, because there are sometimes nucleotide repeats. And basically what happens is that when a double helix comes back together, it might align incorrectly. And if it does, if it binds the wrong complementary base to the opposite strand, then you can end up with a little bubble. So let me show you what I mean by this. So on our top strand, we've got our new strand that is being built by DNA polymerase. We've got our template strand there on the bottom. And we've got this set of 4 adenines and 5 thymines. So let's imagine that our DNA polymerase is moving along and adding in complementary base pairs. Now at this point, we have some strands that are pulled apart, and we've got a lot of movement happening. So if for some reason our strand slips a little bit and one of those A nucleotides essentially gets pushed out and the next A binds across to the T that this one that has bulged out was supposed to be binding to. Since we're replicating, this strand is going to continue replicating and adding in nucleotides as we go. And so what we end up with is a strand that has this A up here on the top, but also because that strand has been shifted just a little bit and replication continues, we end up adding another A in there. And so the result is that this new strand, this new top strand, has an insertion. And so the next time it is replicated and pulled apart, DNA polymerase is going to come along and insert a new base pair there. The opposite can happen. Let's say that on the template strand, that one of those T's pops out and we get a little shift and the complementary base pairing happens with the other strand, and so we get our base pairs lined up. But what happens here is this nucleotide is now sort of bulged out. And in the new strand, instead of having 5 adenines represented, which is what we should have based on our thymines, we end up with only 4. So the template strand maintains that, but now the new strand has a deletion. So our new strand has that deletion. So the next time that it is replicated, we now have a strand that doesn't have one of those thymines and it doesn't get passed on to the next generation. We can also get insertions and deletions by unequal crossing over. We have already seen this. I mentioned that we saw some chromosomal alterations in chapter 6. We were talking about large scale insertions or deletions. We were actually talking about duplications or deletions, where the duplication is the insertion that we see. So we have already talked about this, but again, this happens when there are homologous regions and those homologous regions don't line up quite correctly. So if we look here, we've got AATT, AATT, and our second strand lines up with the wrong AATT. And so on a small scale, when crossing over happens, one of those sets of A's and T's gets transferred to one strand and lost from the other. So we talked about how duplications and deletions essentially happen in tandem. One strand has to lose for the other to gain. The same thing is true for small nucleotide deletions. It is lost from one strand, gained on the other when crossing over occurs. Mutations can also be caused by spontaneous changes. So these are spontaneous replication errors. Sometimes we get a mismatched base put in. And so we then have, say, a purine for a purine. Now we've switched an adenine to a guanine, something like that. This can just happen because everybody makes mistakes, including DNA polymerase. We've talked about this several times. We can also have a spontaneous chemical change. What this means is that a functional group on one of our nucleotides is changed. And there are a couple of examples of this. One is called depurination. This is where the purine ring or the purine bases, the purine nitrogenous base is lost from the nucleotide. And what you're left with is just the phosphate and the sugar. So that guanine in this case is lost, it's broken off, and you're left with a nucleotide that is lacking its base. What this is going to do is essentially confuse DNA polymerase because it's going to get to that and it's not going to know what to insert. And so it may insert something random, which could cause a mutation. Deamination is the same thing. An amino group is an NH group, and these can be lost as well. So for example, if we have a cytosine that loses its nitrogen functional group, we instead have an oxygen in its place. This is going to change a cytosine to a uracil. And so now we have inserted the wrong nucleotide into our molecule. And finally, sometimes outside factors such as chemical exposure or radiation can cause mutations as well. So these are going to basically be environmental factors. These could be things like UV exposure, smoke inhalation, cigarette exposure, things like that. For one example here, we have got radiation-caused dimers. A dimer is where two nucleotides pair side by side instead of across with their complement. So in this sequence here, we've got AT, AT, they should be bound across, but instead UV radiation causes thymine to bind to itself. And this causes a little bump in the structure, jams up DNA polymerase. It can't recognize those thymines correctly because they're bound to one another instead of being bound across to the adenine. And so if this is not fixed, it can lead to mutations in the cell. Also, this is going to interrupt transcription, because RNA polymerase isn't going to be able to recognize this either. So we won't be able to get a messenger RNA from whatever this particular gene is, which means we're not going to get the appropriate phenotype.